Okay, transcription happens when we're trying to make an mRNA molecule. So it's the DNA message being copied onto an RNA strand. Okay, if you look at the diagram, there's sort of a light green kidney bean shaped thing in the middle. That's RNA polymerase, not the same as DNA polymerase. So the first thing that happens is the RNA polymerase attaches to the DNA molecule at a promoter site. I'll explain that. This is the beginning of a gene. Remember that much of your DNA is so-called junk DNA. Not all of your DNA codes for proteins. So the RNA polymerase has to know where to begin, and there's a little secret code in your DNA that tells the RNA polymerase where to begin, and that's called a promoter site. Next, the RNA polymerase starts adding free RNA nucleotides to just one side of the DNA. Remember that when we had DNA polymerase replicating the DNA molecule, the DNA polymerase worked on both sides of the DNA. RNA polymerase only works on one side of the DNA, and it's just making this single-stranded mRNA strand. Okay, the RNA polymerase keeps moving along, attaching one letter after the next. Remember, it's attaching, if there's an A, it attaches a U, if there's a C, it attaches a G, and so forth and so on. It keeps moving until it reaches the termination signal. Basically, it's a signal in the DNA that says, here the gene is done. The rest beyond this is junk, quote unquote, junk DNA. All right, the finished mRNA then breaks away from the DNA and it leaves the nucleus. You see the diagram there, and then it heads to a ribosome. A ribosome, again, is going to be the place where the protein is built. Okay, finally, for this part, when we're talking about making the RNA, there's something called the genetic code. The genetic code is combinations of 3 RNA nucleotides that code for specific amino acids. Remember that the RNA is read 3 letters at a time. Each of those 3 letters is called a codon, and they code for one specific amino acid. And there's a table that shows which codons code for which amino acids, and that's on the next page. Okay, on this page is the table for the codons, and you see if you had a codon, let's say, let's just pick one, let's say AAA. So the first letter A is found in the first column on the left, so you find the letter A in the column on the left, and then the second letter A goes across the top. Do you see where it says second base? Find the column A and find where the row and the column hit each other. And then the third letter is off on the far right side, and you find that one line that has AAA. Where those 3 intersect it says AAA, and the amino acid is called lysine. Likewise, if it were AAG, it would also be lysine. There are 64 possible combinations of codons of 3 letters, but there's not that many amino acids. The other thing to be aware of, every mRNA molecule starts with the codon AUG. That's called the start codon, and here it's circled or boxed in green. There are 3 codons that indicate the end of an mRNA molecule, and that's the UAA, the UAG, and the UGA. Those are all called stop codons. That's it. Now what we need to talk about is protein synthesis, also known as translation. That's the next set of notes.